In this video lecture, I will be talking on Western blotting. Western blotting is a technique used to detect and analyze proteins, in which proteins are separated by gel electrophoresis and transfer to a blotting membrane. Then the protein of interest is identified by a specific antibody. So accordingly, Western blotting is also known as immunoblotting. Western blotting started by preparing the sample by using lysate informed reagents, and these include, as can be seen here, sodium dodecyl sulfate and mercaptoethanol. And as can be seen here, the protein becomes linear. This is the protein before adding the lysate. It is in a complex and a tertiary form. Following adding the lysate or the loading buffer or the lysis buffer, including SDS and beta mercaptoethanol, the protein, it becomes, as can be seen here, in a primary form and more linear and coated with negative charges acquired from SDS. Beta mercaptoethanol also play a role as they destruct the double sulfide bond, and the protein, it becomes linear. Then proteins following this process are placed or loaded into the wells on the top of acrylamide gel. As can be seen here, we load this protein. So SDS PAGE or sodium dodecyl or dodecyl sulfate polyacrylamide gel electrophoresis, this is a technique for separation of proteins, and this technique actually separates primarily by mass according to the molecular weight, from the highest molecular weight to the lowest molecular weight. So when the current applied, all SDS bound proteins, these are SDS bound proteins, migrate in the gel towards the positively charged electrodes. Following that, protein bands, these are within the gel, are transferred to a blotting membrane, which could be nitrocellulose or polyvinylidene difluoride, as this membrane have high affinity to protein and they have a retention capacity. Then gel and membranes, as can be seen at this gel and membranes, together with the sponge, on top and bottom and filter paper below and above the sponge are held inside a cassette. This is a gel cassette or gel electrophoresis cassette, where the gel is at the negative electrode. This is the gel as the side of the negative electrode inside the, which are held, these are held inside the cassette, and now they are in the buffer tank. So when current applied, as can be seen here in this, following application of current, protein bands move from gel to the membrane. The binding of protein bands are increased by adding methanol to the buffer or transfer buffer. So very crucial step following that should be taken, which is blocking by means of adding BSA or bovine serum albumin, and this to prevent binding of detecting antibodies from binding to areas in the membrane where there are no bands, interspecies. However, the bands do not get affected or do not get affected by the BSA. So following this, membranes are incubated with primary antibodies. So the antibodies with specificity towards the band will bind, and this followed by washing to remove unbound antibodies. Then secondary labeled antibodies will be added. Usually the secondary antibodies, labeled antibodies, they are bound or labeled with horseradish peroxidase, which colors the target protein into a purple following adding substrate, and now the protein band of interest will become visible. Thank you very much, Professor Ahmed Bolad.